Yes.
Okay, hi everybody. My name's Trevor Pugh. I'm a senior scientist at the Princess Margaret Cancer Center, coming to you from Toronto. As well, I direct the genomics program at the Ontario Institute for Cancer Research. Part of my role is overseeing a core that offers single-cell sequencing at the Princess Margaret. So I'm going to be really sharing some of the technologies around generation of single-cell data, as well as sharing some research examples where we use conventional droplet-based single-cell sequencing, nuclei-based sequencing, as well as spatial RNA transcriptomics, and really starting to weave some of these technologies together. These slides were all covered by the same license as the rest of the CBW. So the learning objectives for this session are to understand the conceptual shift, moving from bulk RNA sequencing to single cell. There's really some technical considerations, especially as you try to make sense of that data. Learn the different types of various single cell technologies. You've likely, if you're in this workshop, you've probably encountered many of them. Some of them are very marketing heavy. Some are a lot of academic literature. So really getting into some of the details as to what these technologies are. And then I'm going to share some examples from my field, which is cancer research. So using cancer as an example, really illustrating some of the types of questions you can try to answer using single cell, and then talk about some of the forward-looking applications of single cell technologies. Are there any questions in either Ottawa or Edmonton about the scope of this opening session? Okay, hearing none. I'll move on. So the first fundamental concept is we're all made of cells. So the unit of analysis here really is the cell, and tissues and tumors are not these homogeneous mixes. They're actually highly complex mixtures of immune cells, stromal cells, and many other cancer types. So it's not just a bowl of M&Ms, it's actually a very nicely and importantly structured series of cells. And this is really one of the first assumptions to consider when you're looking at the technology. Do you care about that structure? Do you actually have to have the spatial information? Or are you really just interested in quantity of cells, proportions of cells, and trying to infer cell-cell interactions without knowledge of the structure? And for a lot of the scientific questions you want to answer, the structure really may not matter. So I really would caution groups from just trying to jump on to the latest spatial transcriptomic methods, where conventional droplet-based methods may be totally fine to achieve your scientific goals. I'd also put forward single cell analysis is not new. We've been able to do single cell work for a very long time. Most famously, karyotypes. This is my karyotype. This is one of my blood cells dropped on a glass slide. So we've been looking at these as cytogeneticists and molecular geneticists for a long time. In situ hybridization of a single transcript is a very common diagnostic test, even today. But the real revolution in single cell now is the scale, the completeness, not just looking at one transcript like in the lower left, but being able to really paint and look at every single transcript in every single cell in a tissue section. And really that ability to not just look at one set of chromosomes from one cell, but to look at all the chromosomes from every single cell. So in the upper right here is an inferred copy number track from a cancer sample, and on the lower right is a spatial transcriptomic experiment, really looking at hundreds of transcripts or hundreds of genes that are expressed in transcripts across an entire cross-section of a tissue. And this is really the realm that this opening talk is really all about. Single cell analysis, the real secret here is it reveals heterogeneity that gets masked when you do conventional bulk sequencing. So if you took these 6 toy cells on the left-hand side, ground them up, and sequenced their bulk RNA, these 3 toy transcripts would all look like they're expressed at the same level. But if you have single cell information, you can really start to look at how individual transcripts differ across different cell types and across different cells within a sample. I put the title of this slide in quotes, is actually a quote directly out of this really well-aging review. So this is a review of all the single cell technologies back in 2019. It actually hasn't changed very much since then. Technology kind of took off, and now it's more iterations on these core sets of technologies. I've labeled across the top sort of the three broad buckets where a lot of single cell work has been focused. One is in lineage, so going from a stem cell and trying to understand how cells develop, both in normal tissue developmental processes, but also in cancer. And here's some of the foundational bioinformatics tools to do that. Cell state, so just trying to understand what is a cell, what functional state is it in? If it's a T cell, is it exhausted? Are there gene expression profiles associated with not just the label of the cell, but also its functional state? Tons of ways to do this. You can look at the DNA, you can look at the RNA, you can do chip at the single cell level. We're going to focus on this one little box here. We're going to focus on RNA sequencing, but just to know there's a whole world of single cell measurements that you can use to validate or to integrate with your RNA sequencing data. And then trajectory, trying to look at cells within a tissue, or in my world, in a tumor, and trying to understand what is the trajectory of cells, either trajectory within a developmental process, potentially in a mouse model, a zebrafish model, or even within a tissue itself. Is there a stem cell at one side that's kind of giving rise to other cell types? So I want to talk about some of the considerations and capabilities when you generate your data. So what do you have to think about when you're trying to design this experiment? And especially even as computational biologists, it's really important to be very involved in the wet lab and the design of the wet lab experiments, thinking very early about the controls. It's very hard to kind of inherit data where you weren't really fundamentally involved in the laboratory and the design of the laboratory experiments as well. I put this quote, this title in quotes as well. It's also taken from the abstract of this paper. And really, it's just showing this really upward trajectory. Even though this stopped in 2018, it actually just continues to scale. Really, the scaling of just the pure number of cells that you can isolate and sequence from a single sample, starting with a single tube. So literally one cell in one Eppendorf tube, moving to plates, moving to microfluidics, ramping this up with high throughput robotics, nano droplets. This is really what the 10x genomics platform is based on, and so on. So really, we can sort of rely that there's going to be this continued increase in the number of cells that we can affordably profile in a single experiment. The devil is really in the details, not so much in the box that you use for the single cell work. It's really how do you collect and process your samples. And this is probably the number one predictor of success of a single cell experiment, is really thinking of all that upfront work from the organism, be it cell culture, mouse, human on this slide. How do you actually get your single cells? Are those cells viable? Are they coming through your flow cytometer, or however you're isolating the cells in an intact way? And then how do they get sequenced? So really the importance of building in quality control checks to really score out the effects of tissue digestion, molecular barcoding, if your method includes it, and the sequencing. Have you sequenced deep enough? Have you saturated your entire sample? Are you detecting every single transcript of your sample? Or are you just looking at the top? You're looking at the top 5 or 10%. And really, these parameters, some of the questions I put in here in orange, these parameters really come out in the quality control data. One of the most famous metrics is mitochondrial content. So if you have very high mitochondrial content in one of your cells or across all your cells in the experiment, it's often, but not always, but it's often a sign of degradation. Your cell is essentially dying. You're not actually getting the cytoplasmic RNA. You're just looking at the mitochondria from the intact mitochondria. So sometimes that is an indicator that something has gone wrong upstream of you. There are many ways to isolate single cells. You can do it by hand with a pipette. You can use a microscope. You can just pick them up with a microvet. Flow sorting, certainly in my lab, we've had good experience with this. Laser capture microdissection, literally a laser to cut out those single cells, put them into a tube for your molecular biology. 10x genomics, where I'm going to be spending most of my time, and I think you're going to be working on some 10x data, essentially encapsulating cells within droplets along with your reagents. So you do the reaction within an oil droplet. You can put them into wells. And circulating tumor cells. So actually using magnetic beads to pull out cancer cells from a very complex blood-based mixture. So there are a lot of ways to get single cells. Some of these are more scalable. The manual method's obviously very difficult. 10x having been very successful in using these droplet-based methods to really ramp up the number of cells that you can analyze in a single experiment. The state of the art today is on the order of thousands of cells. So 1,000 to 8,000 is a pretty conventional 10x experiment, although there are ultra deep single cell protocols available now. One my lab's been looking at most recently is called Turtle-seq. Lets you look at tens of millions of single T cells if you're interested in targeted T cell receptor sequencing. So one example where if you have a very narrow focus or very narrow question, you can use these technologies to go extremely deep into cell populations of interest. In some cases, we don't have fresh cells. So in this case, you can actually turn to nuclei, where you can take a fresh or a frozen, even some formalin-fixed paraffin-embedded tissues. You essentially collect your tissue, blow away the cytoplasmic component, isolate the nuclei, which are they're like harder. It's like a harder rock. So you can actually pull out these nuclei, use flow cytometry to pull out the intact nuclei. So when you do that first digestion, some of the nuclei fall apart. So you get this debris. So you use flow sorting to pull them out. And then those nuclei can then go into conventional single cell experiments. I've shown 10x technology here, but it doesn't have to be. You could flow sort them into plates or use some other method to isolate single nuclei as well. This is a very important design decision because it does have some of these impacts I've put here on the slide. So you have lower transcript counts. The RNA has not been spliced. You're actually seeing a lot of your reads are going to map to the intron. Sometimes it's very interesting. If you're interested in fusion transcripts, this is actually a really great way to go because you can intentionally keep the intron and therefore map back the DNA breakpoint. And there's actually some cells that you'll see in fresh tissue that you won't necessarily see in fresh frozen. So when you're digesting a fresh tissue versus if you took the exact same sample, froze half of it, and processed half of it fresh, you'll actually see different cellular components coming through due to bias in the freezing versus the formalin fixation process. Here's a published example of that, looking at a neuroblastoma tumor. So in this case, they took fresh cells and they isolated nuclei. So this is the UMAP, which we'll be spending a lot of time with, labeling cells and labeling nuclei and then co-clustering them together. And you can see by eye, the UMAP with the cells has cells that the nuclei does not have. And conversely, the nuclei plot has cells that the fresh frozen sample doesn't have. And this really jumps out at the stacked bar strip plot down here at the bottom, really showing that there is cellular bias across these two. So if you're really interested in natural killer cells, you may not want to use one of these approaches versus another because these cells, some are dying when they're frozen, but may be intact if you fix them and are studying the nuclei. So another important design decision that you as an analyst really needs to know about, especially if your study actually combines fresh samples or a sample set based on nuclei. It's actually going to be very challenging to integrate and put these two sample types together. I mentioned 10x genomics. It's certainly not the only single cell approach in town, but certainly the most popular. I think they're kind of the market leader currently. There's two commonly employed strategies: 10x genomics end reads, where you basically prime off one end of the transcript, versus SMART-seq2, which gives you the full transcript, and this is done in plates. So the end approach really has this benefit of scale, very cheap per cell. You can do thousands of cells if you have the budget for it. There's protocols to read off either at the end of the transcript, the 3' end or the 5' end. Uses Illumina sequencing, so as sequencing goes down, this approach gets cheaper and cheaper. SMART-seq2 is a little bit different. It's more expensive per cell. The scale, since it's plate-based, is lower than in the droplet-based methods. But if you're really interested in splicing or you want to get full-length transcripts, maybe you want to try to find a fusion product, then the SMART-seq2 plate-based approach is actually is potentially the better approach. And same sort of comment as on my previous slide. It's very important to know in the experimental design which of these two methods were used. So you don't want to be going to look for splicing patterns in an end-biased set of data, like the 10x protocol might give you. You're likely able to find some, but you do have to know that you have this end bias. My next slide sort of shows that direct comparison. So SMART-C2 and 10x Chromium, where you get the exons, where you get multiple exons over time. 10x Chromium really having this end bias. You do get this internal priming because 10x relies on the 3' protocol on poly A priming. So if you have a little string of A's in the middle of your transcript, sometimes you'll get signal there as well. You may want to analyze this signal. You may not. That's a design decision. Do you want to include these internal priming events when you're counting gene expression? It really depends on your analytic approach. There's no really right or wrong answer here. And then the right-hand side of this plot really showing the differences in scale. So the number of cells, the number of genes that you're able to recover. Certainly the 10x approach, not recovering as many expressed genes as the full-length approach, but then you're looking at fewer cells. So it's a design decision. Do you need lots of cells, or do you have a nice homogeneous population that you want to understand at a very deep level?
I'm going to hop in here with a question from the Slack. How can we get full-length capture in SMART-seq 2 when we still use Illumina machines? Are they not still short reads?
It's actually a great segue to this exact slide. So this is actually from PacBio. So this is actually where you actually do have complete transcript information. You still can get short read information from SMART-seq 2 because you still are getting reads across the entire transcript. So the way SMART-seq 2 will work, it'll amplify the whole transcript and then it gets fragmented up. But you can still count up the number of reads that map to each of the exons. But your point is correct. You don't actually know the linkage across those exons. And that's really where long reads like PacBio really would come in, where you have a gene, you have these different differentially expressed exons. With short read sequencing, you're right. It's very hard to know exactly which transcripts contains which exons. But if you have some of these longer read technologies, you get one long read and it actually lets you stitch all these exons together. So yeah, exactly. If you're very interested in very precise mapping of splice variants, then you want to marry that upfront full-length transcript amplification technology with a downstream full-length sequencing technology as well. And you can think of those two steps in single-cell analysis as relatively modular. Yeah, great question. Does that answer what that individual had in mind? Okay, hearing none, I will push on. Just sneaking a little bit beyond RNA sequencing, one of the experiences we had early on was a bit of disappointment where there were very well-established canonical protein-based markers for cellular phenotyping, especially when we worked with immunology groups that had a set of cell-determining markers like these. When we went to sequence them by RNA, those canonical markers weren't always expressed at a very high level or weren't expressed in a homogeneous manner across all the cells in a sample. So this is an example from this Nature Methods paper. We had this, or they had this collection of cells, but you can see when you color individual cells by individual transcripts, they're not all expressing these transcripts at the same level, even within a single population. So this is where this additional single-cell technology called CITE-seq came in. And the way this works is you have your cell, and then you have a set of antibodies against your cell markers of interest. And these antibodies are tethered with a little synthetic DNA sequence. And when you encapsulate your cell with this antibody stuck on the outside inside an oil droplet, so that's marked here in blue, that DNA tag comes along for the ride when you do the library construction. So you can see here, there's the little tags interacting with the reagent molecule, and then it gets amplified along with the rest of the RNA within the cell. So this has the added benefit of if the antibody is stuck, you actually can read it out using DNA sequencing. And that kind of filled in the gaps where you have that marker, you read it by RNA, where sometimes you'll have spotty or almost no performance. If you look at the CD8 marker, but it really shines through using a protein-based marker as well. And that's really the state of the art of genomics in general, is now this multi-omic approaches, not just in single cell, but also conventional cancer genomics, and cell-free DNA have really started to pursue not just DNA approaches, but other orthogonal approaches like protein to boost your signal. So all the concepts I've just talked about, how to capture single cells, how to measure RNA, now have an additional layer of information with the advent of spatial technologies and spatial transcriptomics. Um, I hesitate to call these spatial RNA sequencing because some of these spatial technologies aren't always using sequencing. Sometimes they're just using probes, but they can use hundreds and soon to be thousands of probes to try to look at the entire transcriptome. This is a very beautiful marketing slide from 10x Genomics, but really behind it is just a matrix. It's just a set of cells that are basically a table that tells you the distance from one cell to another. Otherwise, the single cell data really is exactly the same. You have a grid of cells by gene, and then spatial transcriptomics adds another matrix that gives you cells by cells. It says, what is the distance between each of these cells? And it essentially allows you to ask new questions of your single cell RNA sequencing data. So once you've done your single cell sequencing analysis, you now ask questions that sort of sit on top of it. How far are certain cell types from each other? If a cell is expressing this type of pathway, how far is it from another cell that expresses a different pathway? So while the visuals are really quite striking, computationally, it's really just another table. And that table is essentially cell by cell. And then the units of the table are how far are you away from that cell. And then you can reconstruct that table into striking visuals like the one on the left. This is now possible across large pieces of tissue. And actually, the best example I've seen is some example data where they actually put an entire mouse pup, a section of them on a single glass slide. And really the ability to map, in this case, a gene panel across every single cell in a developing organism. So in this case, this is the opportunity to use that distance matrix to look at cell organization in addition to cell state. And this is an example of really all the types of questions that can now be layered on top of single cell data. Tissue development, how do cells change over time within a tissue? In the cancer world, what is the heterogeneity within a tumor or the microenvironment? How's the immune system combating cancer? And tissue architecture, what are the interactions between cells? Are these cancer cells versus immune cells? Are there suppressive programs? Are there activating programs? So really the same questions that you would ask in a non-spatial context, now being able to be asked when you have spatial information as well. And there's a bit of a cottage industry in this cell atlasing activity. I think Gary Bader's going to talk extensively about this, really trying to share all of this single cell data with or without spatial information, but then integrating your own data with the published data as well as important context. This has been very powerful in cancer because we can say, here's a set of cancer sample, a set of cancer cells. How are these cancer cells different from normal tissues, and how are they dispersed throughout a cancer sample? And not to endorse just 10x genomics, there's many strategies to localize a transcript within a cell. So if you have a tissue, cut through your tissue, there's capture-based methods where you basically have a carpet of primers. So you put your tissue down, and then you're actually sequencing and discovering new transcripts. This is more the discovery-based approach. If you know the cells that you're interested in situ hybridization is very effective. You can use a probe for cells of interest, hybridize them to the RNA in the cells, and then map them back to their spatial alignment. And in situ sequencing, where you kind of have the best of both worlds, and you try to amplify every single RNA transcript, map those spatially, and they all kind of land at the same place. You have a table of cells by genes, and you have another table of coordinates of X and Y coordinates. 10x Genomics is a company that has all of these. So there's one here looking at the droplet-based method I sort of opened the talk with. A carpet-based method. This is the Visium approach, and they're increasing the resolution from lower resolution to Visium HD. The challenge with this middle approach is it's not truly single cell. You have a gridded carpet of probes. You put your tissue down, but if your cell happens to land between two probes, it's going to light up for both of those probes. So I sort of consider these carpet-based methods are more like cell averaging. You can see what it's not really truly single cell. It's sort of an average of cells that are in a very small region. And versus the Xenium platform, where you're actually looking at individual cells, you can segment the cytoplasm and say more definitively that a certain transcript belongs within a single cell. But really, all the lessons that we've sort of learned from the droplet-based methods really hold for this sort of carpet-based method as well as the in situ probe-based method as well. And really, there's no right answer. It's really about what is your scientific question? Do you need true single cell information? Is the local neighborhood average sufficient? Or maybe you don't need spatial information at all, and droplet-based methods are totally fine. When I'm asked, I mean, that being said, when I'm asked what technology should I use, I'll often pull up a table like this. And these are kind of the parameters that we think through when trying to choose a technology for a single cell experiment. How many genes do you need? Do you want to sequence every single gene, or do you already know what you're after? And you can actually use a panel that's cheaper and more sensitive. Do you need de novo sequence information? Are you interested in somatic mutations, fusions, splice variants? This sets you down a different set of technologies than if you just cared purely about gene expression level, where a probe-based method might be better. Sensitivity, do you need to get every single transcript in every single cell? Or if you just want to know the cell identity, maybe you only need to look at the top 5 or 10%. These are two different technology or multiple different technologies that sort of balance sensitivity versus breadth. Do you need spatial information? How big is your tissue? If you have a little tiny pinhead piece of tissue, you put multiple on a single slide, or do you have a great big surgical sample where you can do a lot more with it, or you have to choose a certain region of interest? And some of it bakes down to feasibility. Do you have access to tissues? Do you have access to flow cytometry? Is there a genomics facility locally, or is your local group very strong in microscopy? So some of that will also play into sort of your selection of technologies. Okay, so I wanted to just pause there, see if there are any questions about the overview before I dive into a project from my own group where we use some of these technologies in tandem. And yeah, I don't see the Slack, but if there's anything there, happy to pick up those questions.
No questions in the Slack so far. I'm keeping an eye on it. Ottawa, are there any questions here? No, I'm seeing a lot of shaking heads. Edmonton?
No questions so far from Edmonton.
Okay.
Sorry, I didn't.
Oh, we do have one question. You can come on up here if you'd like to ask it directly into the, or if you'd like to put, you'll need to come up to the, yeah, sorry. We don't have a portable microphone.
Question is what's the difference between CITE-seq and AB-seq?
Conceptually, they're very similar. I haven't actually used AB-seq myself, but I think the name implies that both are going to be using a DNA tag tethered to an antibody. So there are a few. This is also where it is challenging to pick up which technology you want to choose. Sometimes there's a marketing term versus the name that's been used in the literature. So I haven't used AB-seq myself, but there are a few different brand names for the CITE-seq protocol. Do you have more information on AB-seq that you could share?
Hang on just a sec.
It's just because one of the postdocs in my lab is using AB-seq, where I think it's a protein probe instead of a DNA, or the oligo is formatted differently. But from my understanding, it's pretty much just a way for her to enrich for T cells instead of using the original single cell the data set that I'm using, because they generated the data I'm using first, and then they did AB-seq because they had some extra money. So, well, I mean, there's more reasons, but that's a pretty big one. But yeah, I wasn't too clear on it. But I can paste a link in the Slack about the like brochure thingy that she sent me.
Okay, yeah, I'll take a look at it. CITE-seq itself does have an antibody, so I just rolled back to that slide. It does have this antibody with the tethered DNA, and the reason for that is then once you encapsulate it in oil, that DNA tag is coming along for the ride. From what you described, they might have been using the antibody to enrich, which is kind of like you would do with flow cytometry, like in like this kind of experiment here. So if you're interested in T cells, they might just be pulling them out using an antibody and then sequencing. Is it the whole RNA? Is it a targeted approach? Those experimental details are super important to understand before you jump into the computational analysis.
Interesting, because we don't use FACS or like the 10x oil-based. We have a collaborator that does that, but we use the BD Rhapsody cartridge for our isolation instead of oil droplets. So
Okay, so that
maybe CITE-seq is just, or maybe AbSeq is just more compatible for that or something.
yeah, it might be a microfluidic chip as well. There's a few ways to sort of engineer that. So I, really worked through exactly the details, specifically of is there a cellular selection and is there transcript selection? So is the antibody pulling out a cell population of interest? That leads you down one road. And are they actually selecting for individual transcripts in some way? And then is an antibody being used to also add in protein signal as well? So there's a few parameters there. But yeah, happy to take a look at the AbSeq protocol as well.
Practically, though, we would see that just if it's pre or post lysing, right? Whether we're labeling with an antibody before lysing cells versus after? Or is there a, is that too simplistic, maybe?
I don't know. I mean, the antibodies will stick with proteins even in a lysate. So
All right.
I don't know if
I'll
you could say
paste
that.
it. Thank you for your thoughts on that.
We do have another comment or question here. Could you come up to the mic?
Okay.
Yeah.
I might be able to add some clarification because I believe one of the projects I work on as well uses Rhapsody-seq, like AbSeq, and I believe they're just like two like very common terms for pretty much the same thing. Because I remember that's the actual protocol name, but they do use like that CITE-seq technology to use like the cellular indexing as well.
Okay, and that happens a lot. They'll take like a published protocol and then you kind of rebadge it. So it sounded like CITE-seq, but yeah, I'll take a look. Thanks for the clarification.
Yeah.
Okay, so I want to share. Oh,
here. Sorry, hang on just one moment. Sorry. We got someone coming up to the front with one more question.
Hello, I'm just wondering if the core facility in Toronto has tested the single cell type by Illumina recently, because it's so much cheaper than 10x, and I'm wondering if they're competitive enough.
Yeah, we're testing it this summer, so we're trying to figure this out. I'm pretty
Oh.
excited by it. The
Okay.
other protocol we're looking at is having running some of the 10x sequencing libraries on an Ultima sequencer. So there's a few routes to cheaper sequencing. I think for me, the most important parameter is sensitivity. Is it
Right.
finding all the transcripts that we find using 10x, either droplet-based or Xenium? So
Will
yeah,
their
coming
will their
soon.
performance metrics be like published for the public to like compare?
We'll probably, and we've done this for other technologies, we'll release just some demo data, either on a cell line or some other sample that we all kind of understand. So yeah, happy to share the data from our own validation experiments.
Perfect.
We
Thank you.
don't usually have huge numbers to publish them, but we usually just release them as like a resource for the community.
Okay, thank you.
Oh, we have another question. This is awesome.
Yeah, so thanks very much. Just a comment on that one. You mentioned about the PeakSeq, right? For the Illumina.
Yeah.
Yeah, it's a PeakSeq. I tested it before and it's pretty much compatible. The only difference is that you don't have to use a Chromium X, which is just making the, yeah. Yeah, but for the T100, it was very similar, but we haven't gone to the deep analysis yet. So that's the comment for your Oh, so actually, it's just little. It's not set in Toronto. I'm in currently McGill, and we are currently setting up for in the Neuron, which is a different kind of institute. But additional question on that: are you going to talk about past biosciences? Any chance the barcode deplexing?
I wasn't planning to go into that in great detail, but I know that platform. Is there something you'd like to know about it?
Yeah, is for when they do the barcoding, have you heard about what's the percentage capture on the cellular level? Because normally in the 10x genomics, you can only capture about 20 to 30% of the those capsules of
Yeah, you go much deeper with parse. Definitely the sensitivity is greater. It's a targeted sequencing approach. Um, it is highly dependent on the quality of your sample. So it's very hard for me to say it's like 1 in a million or 100,000, because I think it's what's happened to the cell before your parse experiment really limits or changes your limit of detection. But we're definitely a fan. The sensitivity is really good. We usually position it for validation experiments. We'll do our discovery with total RNA-seq methods and then go very, very deep using parse.
Have you in your lab, have you tested in the core system in the Toronto, have you tested what's the kind of difference between the parse and the current 10x genomic kits in a way?
We have not done a formal head-to-head because those users have been very different. Because parse, we haven't done total RNA sequencing. It's usually people who have panels where they know what transcripts they're interested in, whereas the 10x users are more in the discovery business. They want to sequence all the RNA and look at all the populations. So I don't see them really as direct competitors. I think the applications are different.
It's a little different. Okay, thank you very much. Thank you.
And I think that's what I've tried to keep with the theme of this talk rather than recommend specific technologies. Just make sure the core concepts are clear in the collection, the processing of your sample, understanding cellular selection, understanding broad total transcriptome sequencing versus deep, more targeted sequencing. Because these are all parameters. There's so many technologies out there. These are all parameters that you can you now have control over when you're designing your experiment. Yeah, these are all great questions. Okay, so in my last 10 minutes here, I will just talk about some of our work in glioblastoma. And really, this starts with the concept that tumors are not just cancer cells, they are cancer cells, immune cells, and many other cells that actually change in frequency and function during treatment. So this work is published. You can read this paper in Nature Cancer. It's cancer stem cell project, really trying to understand the clonal heterogeneity of the stem cell compartment, and then understanding how stem cells are distributed throughout a tumor tissue. The whole concept is around this idea of glioblastoma stem cells. So there's bulk tumor, and embedded within it are these CSC, cancer stem cells. And the problem with chemotherapy is it targets the actively proliferating bulk tumor. And what we wanted to do is understand the cancer stem cells so we could pick those ones out therapeutically and then let the rest of the tumor fade away and prevent regression. So we took bulk tumor. We actually had two collaborators, Sam Weiss in Alberta and Peter Dirks in Ontario. One uses flow sorting to isolate their cells of interest, put them into a plate. The other group uses stem cell culture that selects specifically for stem cells. Both have the goal of arriving at a stem cell population. And then our group's role was single cell RNA sequencing of these brain tumor stem cells. First question we wanted to ask was, are brain tumor stem cells comprised of subpopulations, or are they homogeneous? So are they all one big mass, or are they even at the stem cell level? There's some apparent heterogeneity. And the answer was both. So within each stem cell culture, we found a range. And so these are all the cultures we looked at. They're ordered from monoclonal, and I call them monoclonal because there's two populations. They were genetically the same. One was actively cycling, and one was at a different stage in the cell cycle. So these green and browns are kind of all basically the same clone, but as you sort of scan across this plot, you start to see more and more subclones. And this is the summary. We actually had one that had as many as 6 transcriptional clusters all coexisting within the same culture. So it really showed with single cell sequencing the ability to really zoom in on exactly what's existing in these populations. We could also take that information and infer DNA copy number from the RNA sequencing data. So here you can look at genes that happen to be on the same chromosomal arm. The resolution of this, you can probably get down to a cytoband, but if you look across the entire transcriptome, you could see basically every single cell had overexpression on this arm of chromosome 7. So you sort of scan all the way down. But then this subpopulation here uniquely had underexpression or low expression of chromosome 10. So loss of a known tumor suppressor in glioblastoma. And you can actually play this game over and over. So if you look at this subpopulation, they all had overexpression of chromosome 20. And you could find the sub clone by looking at low expression of this piece of chromosome 6. So this is where you could take the cell assignment for each of these cells, but then also try to explain some of the heterogeneity from the cytogenetic or the DNA alterations as well. The other lesson from this study was the patient's glioblastoma stem cells were all different. So if we took all of our data and tried to cluster them all together, you really couldn't force it. And this is actually now well established in the cancer literature. Every cancer really is transcriptionally distinct. In this case, we had a few cultures from the same tumor, or from the same tumor, sometimes at 2 time points, sometimes 2 different regions in the same tumor. And even those were very difficult to integrate, even though they're actually coming from the same starting point. So we're trying to understand, even though they're transcriptionally distinct, was there common biology across all the stem cells? And actually, it turned out on this plot, the cells were naturally ranked by clusters that were enriched for developmental programs versus cells that were enriched for expressing pathways associated with injury response. And you can kind of see that gradient here, that if you score your principal components by developmental programs versus injury response, these two appeared to suggest a gradient across all these patients.
You have a question.
Oh, sure.
How was sorting in one location and culturing in another location possible?
They weren't from the same tumors. So the tumors in Toronto were done one route, and the tumors in Alberta were done by another route. It wasn't one tumor that we shipped.
We're getting nodding heads. Thank
Okay.
you.
And this is really the story here. When you scored high for one gradient marked in red, you scored low for the others, and then these ones in the middle were quite interesting because they kind of occupied the center of this gradient. And it really showed how important it was to have large numbers of samples for this experiment, so that we had the ability to sample across the entire gradient. And if you plot injury versus development, you can kind of see as cells from certain tumors kind of occupy different points on this gradient. And really another way to visualize it, what's the distribution from injury response to development when you subtract one signal from the other? And you can really see, even within a tumor, there's a bit of a distribution. They're not all totally localized at one point. So when we got away from the cultures, now we wanted to go back to the tumors and ask, do we see these same signals in bulk tumors? So these are GSCs. You have these really nice, clean populations that are either development or injury. If you look at other groups' data, you can see the signal is in there, but it's kind of obscured by the bulk tumors. So you sort of get these little peaks that are suggesting that there's a stem cell population in there. But this actually had not been seen before using bulk RNA sequencing, because they just ground up every cell and sequenced everything. So the stem cell signature was really not apparent. And it was only when you went into the purified cell population, found those signatures, that you're then able to go back to the bulk, even the bulk single cell data, and start to pick out some of these subtle GSC populations using single cell RNA sequencing. And we then took that exact same data from the bulk tumors. We labeled the stem cell population and the bulk population. And from this, we were able to apply a trajectory analysis, because we're trying to understand, how do you go from that rare stem cell population into the bulk tumor? So using this SC-VELO, this trajectory analysis, we essentially showed that the stem cells were always on this development to injury response axis, but then there was an orthogonal axis about astrocytic maturation. So essentially, these stem cells were fountaining out bulk tumor. So the bulk of the tumor actually didn't look like a stem cell, but they were all coming from this two ends of the gradient towards a common mature astrocyte state. So that's really where you could use a purified stem cell population to understand the stem population, but then use gradient analysis to understand how the bulk tumor fountains out of the stem populations. So that took us to the question, where do the GSCs live? Are they like a starry night and they're all like spinning out all these bulk tumors? Or are they like islands that radiate out a gradient of cells? So this is where we wanted to take the work that we've done in droplet-based sequencing and then apply that understanding to interpretation of spatial data. So this is a glioblastoma from Peter Dirks' labs. It's a piece of glass slide. We actually crammed 2 tumors onto the same slide, so we kind of got a 2-for-1 experiment. I just wanted to focus on this one tumor. This is an H&E slide, so this is what pathologists will look at to stage and understand and diagnose a tumor. So it was a glioblastoma, stage 4, was surgical, so it's nice and big. You get a lot of cells. And then we had a 300 gene panel that we designed based on the results of our droplet-based methods. So we did the droplet-based experiment. That was pure discovery, looking at every single transcript. Then once we decided, okay, these 300 genes are important for understanding the gradient, let's apply that to a spatial experiment where we would look at more cells, but within their spatial context, specifically with this question in mind. So here's the exact same image, but now we applied the panel. And this is more of a QC plot, which I actually really like QC. It's really important to look at what your sample looks like before you dive into the single cell analysis. So here, just looking at how many cells do you have? So 200,000 cells, way bigger scale than we could do with droplet-based methods, but far fewer transcripts. We're only looking at 200. How many of your transcripts are within cells? If they're leaky, if the sample is low quality, this number is going to drop. What's the cell density? Is there evidence of over or under segmentation? And then how many genes represent those transcripts? So you can see here really very few transcripts per cell. This is kind of a feature of the platform that we chose. So every gene expressing, 2 or 3 times per cell. And then these distributions. What's your size distribution? Tumor cells are usually the larger ones, and the immune cells are generally small. And then what's your distribution of genes per cell? Kind of a nice normal distribution, some of them expressing more than others. Generally, the immune cells, fewer genes, are more highly differentiated, and the cancer cells are sometimes more complex. So same sort of data, keeping the spatial information the same, taking the RNA sequencing data and then treating as if it was a droplet-based experiment and generating a UMAP. So we kind of threw out the spatial for this first look. Oh, there's about to be a fire alarm here, but hopefully I can make it through. They kicked me out. So using just the RNA sequencing data, trying to label the cells within the sample totally independent of spatial. And so looking at some of these marker genes, we could say the cells marked in green are likely immune. These cells marked in blue and purple are likely blood vessels. And then this large population are all the cancer cells that are clustering together and are very distinct from the infiltrating immune cells. The problem with this approach is you can see for our fundamental question of where are the stem cells, this is very messy, especially this assignment of the tumor cells. So this is where we wanted to bring in the spatial information alongside the RNA sequencing.
We have a question from the slide previous to that. Was this a probe-based approach or sequencing?
That's right. This is a 300 gene, 300 a probe panel targeting 300 genes. We had multiple probes for genes. So yeah, this is targeted. And we selected those genes based on the results of our droplet-based experiment. So to tidy up this data, we brought in that spatial matrix and we said, okay, give us the same clusters, but this time use the spatial information to guide some of the clustering. So this is an algorithm called Banksy, building aggregates with a neighborhood kernel and spatial yardstick. I think that's actually 2024, not 2000. And you can see just by eye how much cleaner these clusters are, but then also the presence of embedded cells within each population as well. So this is really the state of this analysis. The immune cells, really a nice, relatively homogeneous population. You can kind of pick out by eye the blood population. So there's blood vessels that are going through some of these tumors. So you can actually pick out these immune cells that are occupying the blood vessel component. You can see some of the immune infiltration. But really, it showed to us the importance of having some of that spatial information to guide clustering of the RNA sequencing data. And really, this is still ongoing work. We're trying to differentiate stem from bulk tumor cell clusters at the RNA sequencing level, score every single cell so we can do that trajectory analysis to try and understand not just developmental trajectory, but also the spatial location. And ultimately, our therapeutic goal is to understand if there's dependencies between the stem population and either other cancer cell or even normal cells that are adjacent to them. And that's actually the end goal of this project, is try to disrupt the biology specifically of the stem cells. And this is all work done by a postdoc with Gary Bader and I, who's actually organized a spatial RNA sequencing workshop. People are interested in going to that through Canadian Bioinformatics. And yeah, this is my last slide. I'm sure you've appreciated there's a plethora of technologies that are allowing you to look at single cells in different ways. You can measure the exact same biology using different methods. Sometimes methods, as we heard in the questions, different method names for the exact same scientific concept. AbSeq versus CITE-seq, probably a good example. And so really tailor your experimental approach to the scientific questions that you want to ask. Multiple cellular components can be queried from a sample, so you can look at both immune and cancer cells. So work in my lab, at least, we've biased towards sequencing everything rather than trying to pick out a specific cell population of interest, because we have interest in both cancer immunology and cancer cells. And then consider spatial technologies. It's really undergoing sort of a renaissance right now, but really understanding those technical details of how your sample's collected, how you selected for cells, are you selecting for transcripts, and then use that understanding to sculpt your computational approaches as well. So I've shown a lot of references throughout this talk. So this last slide is kind of a collection of all of those. If you're doing brain work, we actually have some funding through Brain Canada to give discounts for brain-specific work. We actually have a single-cell working group, Panoramics of Vision. So if you'd like to join that, that's a computational working group where there's regular speakers, and it's really a trainee-driven community, really helping people understand their single-cell data.